Hello, everybody. My name is Pieter from Bokhoven, and I'm going to give a presentation today about a paper we have published recently about the Alzheimer's disease drug development landscape. So I'm going to share my screen. There you go. So this is the topic of the paper. As I said, this is about the Alzheimer's disease drug development landscape. That we have published recently, I think in the end of last year. And maybe to provide some background, so historically, drug development for Alzheimer's has been really very difficult over the last actually couple of decades or so. So basically most drug development programs have failed, and they often fail in a clinical stage. And therefore, people have argued that the Alzheimer's disease drug development landscape, and in particular the targets that people aim for, so the drug targets, which are often proteins in the brain, that this landscape should really diversify in order to be successful. And this is really much needed. And what we did here in this study, we investigated how the Alzheimer's disease drug landscape is actually evolving over time. And then in particular regarding these targets that are aimed for, and also the treatment modalities that are used. So of course, traditionally, people have been developing a lot of small molecules, but nowadays people use antibodies or even gene therapy also to try and treat Alzheimer's disease. So we have also analyzed that. So maybe to provide some background about the disease. Historically, Alzheimer's disease has been defined by a particular pathology, being the accumulation of amyloid protein aggregates in the human brain and the aggregation of a protein called tau. And it had been hypothesized that this amyloid is actually causing Alzheimer's disease. So by the buildup of these amyloid proteins, you then get stress in the brain, and that leads to tau accumulation, and that then leads to the dying of neurons, and that leads to dementia. And this was a hypothesis that was very novel in the 90s, but this is really heavily outdated nowadays. And if you look here on the right of my screen, you see genetic risk factors of Alzheimer's disease. And what you can see is that there are indeed genetic risk factors related to amyloid beta and amyloid beta production. But apart from that, all other genetic risk factors hint in different directions of what is actually causal of Alzheimer's disease. And therefore, that's actually one of the reasons why people have argued that really you should aim for different type of targets, not only amyloid, but also other drug targets, which is something that we have investigated in this study. So the study that we have performed was for a large part, we took inspiration from a study by Jeffrey Cummings, who is a very well-known Alzheimer neurologist from the United States. And he has published a series of papers describing all drugs that are currently in clinical development. And he divided it by development phase, so phase 1, 2, or 3 of clinical development, and also what types of drugs are in development. And he published a series of papers. By doing that, he also was able to sort of see how this landscape was evolving over time, which is super interesting. But what we have now tried to do in this study is to make a prediction on how this landscape will evolve in the coming years. So how this is going to evolve in the future. And the way we did that is by looking at trends in how the landscape is evolving by comparing drugs in preclinical development to drugs in clinical development. And by doing that, you will see what drugs are currently in preclinical development and that will likely enter the clinic in the coming years. And so by comparing those two development stages, you sort of get a peek into the future of how the clinical development landscape will look in future years. That is the goal of this study. So we had a specific approach. So what we did is we analyzed preclinical drugs in preclinical development. And we did that by analyzing basically public databases of drugs that are in the development for Alzheimer's disease and Alzheimer's disease only. And what we did here is that we selected only drugs that are in development by industry. And the reason we did that was because if drugs are in development by industry, this means that people are actually investing money in these drugs. Well, if you would look at the entire preclinical landscape, with also drugs that are for example, in development in academia, you also get a lot of noise in your data. And we sort of wanted to filter out this noise by looking only at drugs in development by industry. And for every single drug, we checked by a team of raters, so 6 raters, if a program was still active within the last 5 years, also to make the data set accurate and also up to date. So we compared these preclinical drugs to clinical drugs, and therefore we have, for the clinical drugs, we have analyzed all drugs in different clinical trial registries. So of course, clinical trials that go from the United States, but we also looked at registries from Europe, Asia, and also Australia, because a lot of early phase studies nowadays take place in Australia as well. And yeah, often to be noted is that we have only analyzed clinical trials that are currently active. So they have likely started within the last, say, 3 to 5 years. So all these drugs in development, both in the preclinic as well as in the clinic, they are scored by their target class, and therefore we used the CADRO classification system, which is basically a classification system for targets that are often aimed for and are relevant for disease pathology in Alzheimer's disease, which was very helpful actually. So here you see an overview of these CADRO target classes. I won't go into detail to all, but you see the most obvious classes here, so amyloid and tau, but you also see apolipoprotein E, which is a very strong genetic risk factor for Alzheimer's disease. For example, you also see inflammation, oxidative stress, growth factors and hormones. Yeah, you can read through this list yourself, but we have scored all our drugs based on these target classes. So, and these target classes are, of course, often related to protein targets in a human brain. Second, we scored drugs by treatment modality. So whether it's a small molecule or an antibody or a gene therapy or based on DNA or RNA, or for example, nowadays people also develop cell therapies for Alzheimer's disease. Combination products are in development. So we have scored all drugs by treatment modality, and by treatment modality I mean the chemical structure of the active ingredient. So that's we have only looked at the active ingredient of a drug that is in development. So here you see, I think, the most important graph of our manuscript and also of our study. So on the right-hand side, you see a graph that shows all target classes. And then per target class, the amount of drugs that are in development, either in the preclinic, so in lighter color, or in clinical development. And the drugs in clinical development are indicated by the darker color. And what is important to note here is that all drugs in preclinical development are shown as a percentage of all drugs in preclinical development, and all drugs in clinical development are shown as a percentage of all drugs in clinical development. And this is because all the drugs that we have found in preclinical and clinical development were not exactly the same. So to be able to compare trends, we show them as a percentage. So basically these numbers are normalized by development stage. So of course, so here in this graph, it's maybe a little bit difficult to read. So I'm going to zoom in on both the top part of this graph and the lower part of this graph, and explain a little bit further. So what you see in the top part of this graph is that, as expected, you see that neurotransmitters and receptors are less and less targeted in preclinical stages of development compared to clinical stages. And this is really something that you expect, because traditionally these are targeted for Alzheimer's disease. For example, if you look at acetylcholine inhibitors or other treatments that are currently on the market, they target these neurotransmitters. But that's a little bit old school, so to speak. So these are less and less in development, and you see that also in a decrease in these numbers in preclinical development. What is very surprising to us is that if you look at the number of drugs that target amyloid beta, you see more drugs in preclinical development as compared to clinical development. And this is very surprising, because people have been arguing to really diversify also away from targeting only amyloid. But you see here that even still in preclinical development, there are more drugs targeting amyloid beta than in the clinic. So you won't see a decrease in amyloid targeting drugs in the coming years, but even we expect to see an increase, which is really surprising. And the same holds true for tau. Another important finding is something that was also expected, is that inflammation is also an important target in preclinical development and also in clinical development, because inflammation has been shown to be very important in the processes leading to Alzheimer's disease. Now, this is maybe the second most important part of our results, is that if you look at the preclinical development, you see the emergence of a couple of novel target classes. And these are, for example, ApoE and lipids as target classes. So proteostasis and proteinopathies, and lysosomal and endosomal and autophagy. And these are actually targets that are related to really the genetic risk factors of sporadic Alzheimer's disease. So you really see the emergence of target classes linked to the genetics of the disease. And I think that is really very interesting. So, for example, if you look at all the genetic risk factors here for Alzheimer's disease, you see that most in blue are really related to either cholesterol metabolism, of course, APP processing and amyloid beta, of course, that's the strongest genetic risk factor, and the cytosis and the immune response. You see that in yellow. So you really see that people are really in the preclinic aiming at genetic risk factors for Alzheimer's disease. So now, finally, also some results on the treatment modalities that are in development for Alzheimer's disease, and you really see that the more traditional modalities, such as small molecules, are used for these novel types of targets that you see here below, while for the more traditional targets, people are now developing more novel types of interventions, such as cell therapies, antibody therapies, but also gene therapies. You see that here, for example, for amyloid beta and tau, which I think is also an interesting conclusion. So people are only taking risk on either a target class or a treatment modality, but not on both. So here we come to our most important conclusions of our study. So we can conclude that the AD drug development pipeline is really diversifying in terms of targets. And people are now more and more aiming at genetic targets or targets related to the genetics of Alzheimer's disease, which I think is very good. But also still amyloid beta is still a very prominent target class that people are still aiming for, also in preclinical development. So yeah, we will see also in future years, in the coming years, that this will still be an important aspect for drug development and drug trials for Alzheimer's disease. And finally, I want to also conclude that because this this drug development pipeline is diversifying also in regards of targets, we also need to start thinking in the field of developing companion diagnostics that are related to these novel mechanisms of these drug targets. And because in the end, you want to be able to show whether a drug that is aiming for a specific target is efficacious. So you should also develop a companion diagnostics that allow you to show a pharmacodynamic response, whether you actually hit the target in your clinical trials and in your clinical development first. But second, also biomarkers that show efficacy in these drug trials related to this target. So you really need to understand the pathways related to these targets and also develop biomarkers that can go along with this clinical development. And then finally, I want to end with a little bit of shameless self-promotion, because this is something that we are actually doing here at our institute at Amsterdam Neuroscience. So we have a drug development center called the Candidate Center, where we develop drugs related mostly to genetic targets of Alzheimer's disease, but also other CNS indications. And here, importantly, for every single program that we have in development, we also develop companion diagnostics and biomarkers in parallel. And this is, in the end, of course, very important, that once your compound that has been developed or drug that has been developed enters the clinic, you also have biomarkers that allow you to stratify patient populations based on these biomarkers, but also allow you to show treatment response in these patients. And this is super important, and this is something that we are very much focused at our institute. So lastly, I want to thank, well, a lot of people, but mostly my colleagues. So this is the team that has worked on this study. So my colleagues, Priska and Sasha and Lisa, and also my colleagues Arno and Philip that are at the LSP Venture Fund. So they are at the LSP Dementia Fund, and they are very important also in our ecosystem of drug development here in Amsterdam. Jeffrey Cummings, who is really brilliant also in terms of providing input and feedback to the paper. And if you're ever writing a paper, have him as a co-author, because he provides fantastic feedback. And my colleague Jort Vijverberg, who is a neurologist at our center, and he's a principal investigator of many of the drug trials that we do. So thank you very much, and also thank you to the organizers of this webinar. And I'm looking forward to your feedback. If you have any questions or comments, please send me an email at this email address here below, and I would be happy to answer them further. Thank you very much, and goodbye.